So I mainly work on to find out the role of phase separation in secretory granule biogenesis, which I will talk today. So it is now evident from last 15 years that phase separation play a huge role in membraneless compartmentalization inside cells. But the interesting property is the macroscopic material property of the condensate can vary widely, and it based on functional demands, and sometimes if it is not controlled, it has a huge pathological implications. Now while condensates can show extremely liquid like property like fusion, surface weighting and other things, they can also form gelation and amorphous solids, and on the other hand they can also form very structured and very ordered structures like amyloid. I have shown at the right side. So on top of that, upon phase separation, liquid like condensate can also change its material property with time, and there are various proposed mechanism for that. There might be lot of polymer concentrated inside the condensate, so there can be a polymer entanglement, and because of that there might be amorphous solid formation. There can be physical cross-linking and network-like structure formation. Because of that there can be percolation, gelation and lot of things. And sometimes, since a huge amount of protein is concentrated inside the condensates, the protein can now forced to go in the misfolded conformation. There might be a conformational change associated with the phase separation, and now certain interactions can drive for the fibrillation. If all these things are regulated by cells, then it is fine, and cell has actually utilized a lot of condensates which are not very liquid like, they are like solid like in nature. They have used for like storage purpose and other things. But if it is the process is not regulated, as I have mentioned, the fibrillation has lot of pathological connections, and it has already been seen several neurodegenerative associated proteins, they form fibrils. Now I am interested about how hormones are sorted and stored inside secretory granules. So hormones are a class of peptides, proteins. They are chemical messenger. They release into the blood with some chemical message when it is only required to the body. For example, now I am giving a talk, I may be very excited, so adrenaline and dopamine might be rich with those hormones, and when I will be sleeping, I will be rich with melatonin. So when the hormone is required to secrete, then only it will be secreted, and other time it will be stored inside secretory granules in the cell, certain cell types called neuroendocrine cell, which has the machinery for the regulated secretory pathway. So the cell has a basal like the default mechanism which is called constitutive secretion, where protein is synthesized and released. But certain cell types, for example neuroendocrine cell lines, they have the machinery which will package hormone from the trans-Golgi network inside a membranous granules, and which appears as a dense core when it is looked under microscope. So these are prolactin granules. They form a dense core and they store for a long time. So on the right hand side, it is a readily releasable pool of the secretory granules, and when there is information from secretagogue, it will release that. And although they are in the aggregated state in the inside secretory granules, when it will be released, it will be released as functional monomer to serve the function. Now the thing is how hormones are sorted from the trans-Golgi. Several previous reports have mentioned that they have aggregation properties. So aggregation might be one of the mechanism for hormone sorting inside secretory granules. And my supervisor, Professor Mukherjee, he has done one of his postdocs. He has seen that he chose like 42 different hormones, and he found like all of them has a property to undergo functional amyloid if a certain condition is given. Not only that, when immunohistochemistry was performed on pituitary tissue, it has been seen that hormones, they are actually co-localized with amyloid specific dithiaflavinase. So which is kind of giving the information that okay, hormones might form like it is forming functional amyloid. So now I ask the question like how aggregation and related to functional amyloid formation, how it is happening, what is the mechanism for concentrating hormone so that it can form aggregates. So, when we ask if phase separation can serve the purpose to concentrate it in a confined volume to promote the aggregation. So, I have chosen 10 different hormones and they are actually very different from their location. They are very different on the amino acid length, their molecular weight and their secondary structure, and I wanted to see what is happening. So, now, these are the 10 hormones: galanin, somatostatin, beta endorphin, calcitonin, neurotensin, glucagon like peptides, then leptin, growth hormone, prolactin. pH play a very important role in the regulated secretory pathway because pH in the endoplasmic reticulum gradually goes down till trans-Golgi network, and inside the secretory granule pH even goes around 5.5 to 5.8. So, acidification is one of the major thing that happens in the secretory pathway. So, I wanted to check what is happening if I decrease the pH. So, I took all the hormones at a fixed concentration and then I employed to check what is happening in pH 6. So, out of 10 hormones, only 3 hormones I have seen undergoing phase separation. And then when I added glycosaminoglycan glycan, now glycosaminoglycan glycan is called like helper molecule. It promotes protein aggregation and it is also known to be present in the trans-Golgi network along with other metal ions. So, when I have checked the effect of glycosaminoglycan glycan heparin, I have seen all of them undergoing phase separation in pH 6. They have beautiful surface weighting property and fusion property. So, there is a video of fusion, you will see like neurotensin undergoing phase separation, and there is also surface weighting property which is showing very liquid like nature of the hormones upon phase separation. So, now I picked 2 hormones, growth hormone and prolactin. I recombinantly express and purify it for in large volume to further understand what is happening. So, these two, the advantage of these 2 hormones are there is no processing event throughout this hormone packaging. So, several hormones undergo processing like from the pre-pro hormone to pro hormone and other processing. These 2 small peptides, 191 amino acid and 199 amino acid residue, they don't have any processing event. They are very structured. There is hardly any intrinsically disordered region, as I have done in some in silico analysis to find if there is some intrinsic disordered region, there barely any intrinsic disordered region. They have some amyloid promoting sequence when I have analyzed with Tango. So, then I checked if they are undergoing phase separation if I vary the concentration of both the protein. So, if I increase the concentration of growth hormone or prolactin with increasing GAG concentration, they undergo phase separation. But the nature of the phase regime of growth hormone prolactin is quite different. Prolactin mainly undergo phase separation when it is added in the 1:1 ratio, and with increasing the concentration of the heparin, it is undergoing some clumping of the condensates, while growth hormone actually separating very well with increasing GAG concentration. So, that means that although hormones are undergoing phase separation in presence of GAGs does not mean that the interaction nature will be same for every hormones. Also, like we have checked like how heparin is distributed inside the condensate by labeled heparin, and we have seen like for prolactin it is mainly concentrated at the center of the condensate, while growth hormone it is distributed. Also, the size of the condensate is very different if we increase the GAG concentration. For growth hormone, increasing GAG concentration, size of the condensate increase, while prolactin it is mainly at the 1:1 ratio, they are more pronounced phase separation. So, then I have changed the pH gradually from 7.4 to 6. Now, in 7.4 without GAGs it does not phase separate, both of the hormones they do not phase separate much, but if I decrease the pH even without any external added things, both the hormones intrinsically undergo phase separation. and in pH 6 without anything in 200 micromolar around concentration, both of them undergo phase separation. And also we have seen like very sharp kinetics of the phase separation when we have performed light scattering as compared to its pH 7.4 phase separation kinetics. Also we have performed centrifugation assay to understand what is the dense phase and dilute phase concentration, and we found like in the dense phase the protein is concentrated in the millimolar range, and it is also known previously that inside the secretory granules also hormones are heavily concentrated in millimolar range. So the phase separation might be one of the mechanism for concentrating a huge amount of hormone inside secretory granules. Then we further employed the effect of chondroitin sulfate, another GAGs, and effect of zinc metal ion to understand how the phase regime is changing with further addition of these two parameters in pH 6. And we have found like growth hormones, specifically in presence of zinc, they have formed some clumping type of nature and clump condensation, while prolactin in presence of CSA they formed condensates, and the effect of clumping in presence of zinc is less in prolactin as compared to growth hormone. So growth hormone has some kind of special behavior with zinc, we will see what is happening, and then it is so far so good that okay, they are undergoing phase separation. What is happening after phase separation? So we have done FRAP experiment. So what we have done, we bleach a certain region of interest, and we have monitored like how that region is recovering with time. So it is one of the way to understand the translational dynamics of the molecule. So if the molecular translational dynamics of the molecule is very high, the FRAP recovery will be very fast. If it is low compared to our detection rate, then the FRAP recovery will be low. And it is one of the way to understand, okay, there is some kind of maybe structure formation or some network or some cross linking, something is happening, that is why the FRAP recovery is not like happening fast. So we have seen like all the hormones at 0 hour compared to 0 hour, within 1 hour the recovery is very low. We have calculated the bromide fraction at 0 hour and 1 hour. We have seen like all the hormones, their FRAP recovery is lower at within 1 hour. Then we further look in details what is happening for growth hormone and prolactin. This is pH 7.4 phase separation in presence of GAGs, and the initial recovery while around is 80%. In pH 6, the initial recovery falls around 40%. So pH is promoting some kind of solidification inside the condensates, and it is true for both growth hormones and prolactin. So we have checked the how this liquid to solid transition happening. Now the next question is, what is the solid? Is it amyloid? It is what is this? So we have tracked with one of the amyloid specific dye, thioflavinase, of all the condensates, and we have imaged at 0 hour of phase separation and around the 12 hour, and we have found like all the hormones there is thioflavinase fluorescence enhancement, and which is very predominant for certain hormones. So for example, galanin, somatostatin, beta-endorphin, their thioflavinase fluorescence enhancement is very sharp. And we have also seen, like you will see that fibrillar structures are emerging from the condensates, and there are these 3 candidates which are showing very sharp thioflavinase fluorescence enhancement with time. And there are some other beautiful image of how fibrils are emerging from the condensates, and these are thioflavin TEM electron microscopy image of the condensates along with the aggregated structures. Now this is thioflavin TEM images for 5 candidates, hormone candidates, and we have found that at 12 hours they are mainly the mostly aggregated structures. We have also performed FTIR to understand the secondary structures, and we have found like for example for galanin that 1695 peak is associated with the cross beta structure, and you will see like with time it is increasing. Around 12 hours it is very much increasing, and all the like here the under study all the candidates, they have that cross beta secondary structure which is shown by this characteristic peak of 1695. So then we try to understand what is, because we have the control for growth hormone prolactin, we have huge amount of hormone, and we have found like it is phase separating in pH 7.4, in 6, also in presence of GAGs, metal ions. So we employed all the individual conditions, and we have seen like the thioflavin T kinetics in presence of all the conditions. We have seen like although both the hormone undergo phase separation in all the conditions, certain specific condition is required for the thioflavin T enhancement, and for growth hormone it is zinc, which is showing the thioflavin T enhancement, and for prolactin it is chondroitin sulfate, which is showing the major thioflavin T enhancement. Another study we have performed, we have done time dependent dot blot assay to understand like and we probe the dot blot with amyloid specific antibody OC and oligomer specific antibody A11, and we have found like from the very beginning growth hormone is showing OC positive, but it is negative for A11. So they are actually associated with very fast aggregation, amyloid aggregation with no oligomerization, whereas alpha synuclein, when we have studied, we have found it is alpha synuclein phase separated solution we have employed, and we have found like they are positive for A11, they have oligomeric population, but up to 12 hours there is no amyloid aggregation. So it is showing like growth hormone is undergoing very fast amyloid aggregation in presence of zinc. We have also purified both aggregates from fibrils from both growth hormone prolactin in that specific conditions, and it is showing the XRD showing the characteristics peak for characteristics diffraction pattern, which is equatorial and meridianal for like amyloid, which is 4.4 angstrom and 10 angstrom. And also like we have seen under the electron microscope, it has fibrillar nature. Here is also we have seen like 1695 peak in the FTR, which is showing they have amyloid fibrillar nature. Now, after performing those in vitro experiments, we were interested to see what is happening in the neuroendocrine cell lines. So I have mentioned that certain cell lines like has that special machinery to store secretory granules, and ATT20 is a mouse pituitary cell line which endogenously produce ACTH and beta endorphin hormone. So I have done the immunostaining, and we have found like both the hormones are actually stored in the co-stored in the granules, and they are also showing co-localization with granule specific markers like chromogranin A. So this cell is a good model to study for hormone aggregation. So then when I have transfected transiently growth hormone with EGFP, so the plasmid construct has growth hormone, at the C terminus it has EGFP, so that we can monitor what is happening by EGFP fluorescence. So when we have transfected it, we have found one population at the perinuclear location and one population at the cell tip. Now, perinuclear location, when we have stained with trans-Golgi specific antibody TGN38, it is co-localizing. That means that perinuclear location is actually located in the trans-Golgi region, and they are actually nascent granules, whereas the tip population, they are matured secretory granules and they are ready for the releasing purpose. And now, when we have only transfected with EGFP, there is no granule formation, there is no puncta formation, and whereas the growth hormone, when it is along the EGFP, there is the puncta formation, and that means that the puncta formation is very much protein specific phenomena. Other thing is when we transfected this in a constitutive pathway containing cell line, for example 3T3 fibroblast cell line, we have not seen any puncta formation. That means cell type is also very important to study the how this process is happening. So these are the dynamic nature of the granules. You will see they are highly dynamic in the cell. So this is the TGN population and the tip population, and also to understand the dynamics in the 3D volume, we are doing some lattice light sheet imaging, and it will be it will showing how dynamic they are inside the cells. And also we have found in the TGN fraction those condensates, those TGN fraction is highly packed with the condensates, and they are actually undergoing frequent fusions. Then we have checked that the granule formation at time dependent manner, like at early time point around 16 hours, then 24 hours and 48 hours. We have seen like with the time the granule size is increasing, that might be linked with like more accumulation of matured granules as compared to nascent granules. And also then we have isolated the aggregated fraction and the soluble fractions by treating the cell with lubrol. It is one of the well-known method for secretory granule to isolate the aggregated fractions. We have seen like at early time point like 16 hours, the aggregated fraction, which is the pellet fraction, and here it is noted as P, and the soluble fraction is noted as S. The early time point, the soluble fraction is very high and pellet fraction is low, and with time that soluble fraction is low and that pellet fraction, that means the aggregated fraction, is very high. That is indicating that at later time point, in the since it is a transient transfection, so there is only one time it is treated with that plasmid, and then protein is not stably expressed, and with time there is more population of the mature secretory granules inside the cell. Another experiment we have performed, we wanted to see what is happening by changing the temperature. 5 minutes, yeah, I'll finish quick. So previously it was shown, it was reported that at 20 degrees centigrade there is no mature granule formation. So we thought that might be because of at low temperature the aggregation is now inhibited, and indeed we have seen like when the temperature is low, the lubrol soluble fraction is the pellet fraction is almost nil at low temperature. That means the aggregated fraction is there is not much aggregated fraction, and along with that the cell does not have the tip population. So we have also seen that in 20 degree thioflavinase also binds less as compared to 37 degree. Another thing we have checked, that what is the pH responsive behavior of the granules, because it has to be reversible to release that aggregated fraction in the cytoplasmic pH 7.4. So when we treat the cell with chloroquine, which is known to increase the pH of the acidic organelles, we have seen like destabilization of the granules, and now there is more soluble protein fraction throughout the cell after chloroquine incubation. And also the in vitro study shows that when we increase the pH, the turbidity goes down, so phase separation is reversible with increasing the pH, and again it is when going back to 6 the turbidity is high. So the phase separation behavior is also pH responsive. And now we try to actually differentiate the TGN fraction and tip fraction by fluorescence lifetime, and we have found like that EGFP lifetime is less in tip population, which is showing it might be in the aggregated state. Then we have actually isolated that EGFP fractions, and we have seen like it is positive for amyloid specific antibody OC, and also we have isolated the secretory granule fractions from ATT20 cell, and then we immunoprecipitated that EGFP granule fraction, and they are under the electron microscope, they are granules. We have performed cell cross section study, and in like when the cell is cross sectioned at 50 nanometer, and then we have found like they are forming that granules. And now this is one of the interesting study. So now ATT20 or any neuroendocrine cell, they store that population for release, and that release happens when there is a signal from the secretagogue. So we mimic that secretagogue mediated stimulation with BACL2. When we add BACL2, we have found that the tip population is vanished upon BACL2 addition. That means that portion is now released, and when we actually collected that media after the BACL2 treatment, and we have compared it with the basal treatment, we have found like upon the secretagogue mediated treatment, and that protein is actually releasing growth hormone, and we have actually quantified also that data. This is the last experiment. So what we have done, we have now deleted that amyloid region from the protein. So there are two amyloid region in growth hormone prolactin. One region is in the signal sequence, and signal sequence actually cleaved off when protein enters into the ER lumen, and another protein amyloidogenic sequence is inside the protein throughout the process. We have systematically deleted these two amyloid region from the plasmid and we have checked, like when that amyloid region from the signal sequence is deleted, that protein is pancellular, there is no granule formation. When that region is deleted from the peptide sequence, there is like here and there granule formation, but they are not like informed as TGN fraction and that cell tip fraction as we have seen before. So that means that amyloid sequence has certain information with it, which shows that it has to be formed granules and it has to be released in a systematic way. We have also done some in vitro study where we have seen that when we release that amyloid region from the protein is now in presence of PEG, it is actually in the surface, it is not a proper phase separated, like it is a hollow condensates. So which is showing like the amyloid sequence is very important. And now here I will stop and I will thank my supervisor Professor Maji, and this is our beautiful group and we enjoy a lot. And this is there are other people who have actively worked, Punna, Prasad and Anindo and Sutap. Thank you. I will happy to take questions here, please.
Sandeep. Okay.
I have a comment and a question.
Sure.
Comment is based on the slide in which you showed soluble and pellet and the population is inverting. If you could just go to that slide, I think about 15-20 slides back. Yeah. So look at those surface, the soluble and pellet bands.
Right.
Although the population inverts from 24 hours to 48 hours, but there's one thing that's seen uniformly, which is that the mobility is not the same. The soluble band is always a little above the pellet
Little
band.
above, yes.
What that suggests is that there is some processing going on and the polypeptide chain is not of the same size.
We have actually observed it very consistently.
So there must be, if you do some mass spectrometry
Right.
to see what's happening at the ends,
Right.
there must be some N-terminal or C-terminal processing which is driving it towards the pellet.
That might happen.
So that's the comment.
Thank you for that.
The question is, of what possible use is it for nature to cause the growth hormone to form amyloids in granules and then release those granules? What is the provision for the protein to then go back into solution and work from the amyloid aggregates?
So
how do they dissociate? Is it a pH dependent thing? I mean, is it being kept in an amyloid form at a particular pH and when it's released it dissociates? So what is happening there?
So on contrary to the pathological amyloids, they are actually very responsive, like they are kind of reversible nature. And here also we have seen like with increasing the pH. Right now actually I'm doing the experiments where I have isolated that functional amyloid from growth hormone and I'm checking that with increasing the pH, how that functional change, like secondary structure change is happening. And yet we have observed that with increasing the pH, they are actually dissociated. Like they have that pH reversibility. And previously also in the 2009 paper, it has been shown that when it is like there might be two factors. One is pH and another is dilution, because inside the secretory granule it is heavily crowded, heavily concentrated, and when it is released in the cytoplasm, there is a huge dilution. So dilution is factor number one, and another is pH, which actually makes it reversible.
So that's very interesting. So presumably from the liquid phase separated state also there is reversibility, and from the liquid solid phase separated state also there is.
Yeah. So
what is the kinetic partitioning like? I mean, why are there these two stages, and why do you observe both interconvertibly and then the third interconvertible state, which is the soluble state?
Yeah, very good question, but it is very hard to distinguish between the liquid and the solid because this process is so fast and it's kind of a spectrum which is merged with each other, because within 1 hour we have seen like there are some hormones which have been showing that within 1 hour it has started to bind thioflavinase. So it is like phase separation very immediately, and we don't know how they are they at all different, like different process, or how quickly they are happening? What is the molecular mechanism for that? It is I can't comment on that. Yeah.
Okay, just on this video only. So you have these droplets. Now you saw that they were moving a lot.
Yeah.
Now, why are they moving? Like, is there some sort of a concentration gradient? Or, I mean, I'm just curious, is there some, and what kind of diffusion pattern do you see of the droplets? The second question is, do you see fission also, or you just see fusion?
Very good questions, and I think it is these are some aspects which we should look in, like in future. Right now, I have not actually studied how the diffusion, what is the diffusion property, but actually they are very much dynamic. So there might be some reason behind that, and this is an interesting aspect to look into the future. And fission, I have not actually noticed to look for the fission, and I'll keep that in mind.